Okay, welcome to the AQA A-Level Biology Paper 2. This is the 2022 walkthrough. Let's go. Okay, this paper you got 2 hours to do, and there's 10 questions. It's the 7402 is the code if you want to look this up, and I suggest you pause the video before trying each question. I can't speak. Okay, 1.1. During photosynthesis, plants produce something, compounds which contain carbon, such as carbohydrates, lipids, and proteins. Most of the sugars synthesized by plants are used by the plant in another thing, point two. The rest are used to make other groups of biological molecules. These biological molecules form the biomass of plants. Biomass can be measured in terms of mass of blank 3 per given area of given time. The chemical store in dry biomass can be estimated by using 4, which is the next fill in the blanks. Write the correct biological term beside each number below that matches the space in the passage. Okay, so this is worth 2 marks, so presumably you need to get 2 right for 1 mark. The first space that should be filled in is during photosynthesis, plants produce something compounds which contain carbon, carbohydrates, um, lipids and proteins. Now we call these molecules organic molecules. So the first answer is organic. Now the second answer here says most of the sugars synthesized by plants are used by the plant in, and that'll be respiration. Plants use glucose in lots of different ways to make chlorophyll, to make starch, to make lipids, but mainly glucose is used for respiration. The rest are used to make other groups of biological molecules. These biological molecules form the biomass of plants. Biomass can be measured in terms of the mass of carbon per given area per given time. The chemical energy stored in the dry biomass can be estimated using something called calorimetry or calorimetry. Now, calorimetry is where we burn the biomass under strict conditions, where we try to reduce heat loss and we calculate the rise in the temperature of something like water. Okay, let's move on. 1.2, describe the light dependent reaction of photosynthesis. Sorry, the light independent reaction. Easy to make that mistake. So, light independent means without light. You should by now have this diagram learned off by heart. And if you don't, you need to learn it. So, with this diagram, we have carbon dioxide coming in through the stomata of the plant. Carbon dioxide is then joined with ribulose biphosphate, which we can just write RUBP, especially if you've used the full name once in an answer. Now, there's an enzyme that catalyzes this reaction. It's called the Ribisco enzyme, and it does carbon fixation. It fixes the carbon dioxide with the ribulose biphosphate. Carbon dioxide contains 1 carbon atom, ribulose biphosphate 5. So we now have a 6 carbon molecule that you don't need to know about in AQA. This 6 carbon molecule breaks down to form 2 glycerate 3 phosphate molecules, and we can just write 2 times GP. These molecules are then reduced. They're reduced by using energy from ATP, 2 molecules of ATP, and also using hydrogen and electrons from NADP. Okay, so in order to reduce GP, which happens, it gets turned into TP, which is triose phosphate. Both of these GP and triose phosphate TP are 3 carbon molecules. So you have 2 lots of 3 carbon. Now, they are reduced by adding hydrogen and electrons, which come from NADP, and energy from ATP. Okay, so we need 2 ATP molecules used here and 2 NADP reduced molecules are used. 2 NAD reduced or reduced NAD are turned into NADP, and their hydrogen and electrons go onto the GP and turn it into triose phosphate. Now, what happens to the triose phosphate? What is the fate of the phosphate? One carbon comes off and goes on to make an organic compound like glucose, a lipid for the waxy cuticle of the plant, or storage, carbohydrates such as amylose or amylopectin. But the other 5 carbons are recycled back into ribulose biphosphate. And this recycling into ribulose biphosphate costs another ATP molecule. It costs a bit of energy, okay, to recombine the 5 remaining carbons of the triose phosphate into ribulose biphosphate. And I'm just going to write the number of carbons in each of these molecules here. So those are 3 carbon molecules, the TP and the GP. So that should actually be 3 carbon for each of those. So that is your carbon cycle diagram, not carbon cycle. That is your light independent reaction of photosynthesis diagram that you need to know off by heart. Now, it does say in the mark scheme to this paper that you could score all 6 marks from a diagram like this, okay? Would this diagram get you 6 marks? We may need to say that it's energy from ATP that is used there. If we just show ATP breaking down to ADP plus PI, we've not really told the examiner we understand that energy is released from that reaction. We've not really told the examiner that we understand that when reduced NADP becomes NADP, the glycerate 3 phosphate or GP is reduced to triose phosphate. So I'm going to write out my answer so that you can see what the written version of the answer would look like, but you could go for the diagram as well. I would say memorize the diagram off by heart because that's how I would know exactly what to put in this answer. And just for example, if you were asked how many times would the light independent reaction need to go around to create 1 glucose molecule? Well, glucose is a hexose sugar, so it would need to go around 6 times because each time 1 carbon is broken off of the 2 TP that are formed. Okay, the triose phosphate. Also, the light independent reaction, which doesn't require light, this reaction is also known as the Calvin cycle. So you may see it written as the Calvin cycle in places. Okay, so that's its alternative name. And you need to know where this reaction takes place. And the light independent reaction takes place in the stroma of the chloroplast. So this reaction doesn't take place in the thylakoid membrane because that is where the light-dependent reaction takes place. All right, we're going to move on. This would get you easy peasy 6 marks. Beautiful 6 marks. Lovely AO1 subject knowledge question, this one. Testing you on your photosynthesis knowledge. You should, if you want to get an A star, you should know this content off by heart well before your exam so you can focus on the nuance of exam technique. Okay, moving on to question 2. Put a tick in the box next to the equation that shows how the net production of consumers, N, can be calculated, where I represents the chemical energy stored in ingested food, F represents the chemical energy lost to the environment in feces and urine, and R represents the respiratory losses to the environment. So you've got 4 possible options here. The correct answer is the second one, where we're where we're looking at net production. So the total amount of energy gained is your food, basically, what you've eaten minus what you've pooped out, excretion and urine, and what you've used in respiration, which of course is released as heat and energy for movement and muscle contraction and all of those things. So net production is basically what you ingest minus excretion and respiration. In the UK, some female cattle are only used for breeding. This female breeding herd has dairy cows and beef cows. Table 1 shows the data on dairy cows and beef cows in the UK female breeding herd from December 2013 to December 2017. Pause the video and have a little study through this table. You've got the total number of cows, and then you've got the percentage total of dairy and beef cows. 2.2, in December 2013, the female breeding herd was 48% of all the female cattle in the UK. Use table 1 to calculate the percentage of all the female cattle that were beef cows in the UK in 2017. Okay, so essentially what we're being told here is that 48% of all the female cattle in the UK, 48% of that are female. Okay, and they're shown here in this table, this 48%. We're also shown that 45% of that 48% in 2017, 45% of that 48% were beef cows. What they're asking us then is how many female beef cows are there in the UK? Well, we know there's 48% female cows because it tells us that in the question. And we know that of those 48% female cows, we're told that in 2017 there was 45% were beef cows. So we really need to do 48% times 45%. So 0.48 times 45. And we're going to get 21.6%. You can round that up to 22% if you like. So roughly half of the female cows, which is 48%, were beef cows, or just under half, which equals 22%. And hopefully that all makes sense. Right, let's zing on. 2.3. Use table 1 to calculate the increase in the number of dairy cows in the UK female breeding herd between 2013 and 2017. Show you're working. So we're looking at the increase in the female herd between 2013 and 17 in the number of dairy cows. So let's go. 2013, 54% of all the dairy cows, all the female cows were dairy cows. So we need to do 0.54 times 3.35 million. There was a total of 3.35 million female cows, and 54% were dairy cows. So let's work out the number of dairy cows. And it's like 1.809 mil, right? So you're going to get 3 extra zeros there. Can't be bothered to write it. 2017, we got 3.45 million. So we've had an increase in the total number of females, but the percentage has also gone up. 55% were dairy cows. So we want 0.55 times 3.45 million. And this is going to give us something like, it's still going to be 1 million. So put that in the calculator. It's going to be 1.897 million. Okay, so all we need to do now is find the difference between these two. So take the big number, subtract the small number, and you will get, so 1.897 million minus 1.809 million, and you're going to get something in the 80,000s. Let's whack that into the calculator and see what the increase in the number of female dairy cows was between 2013 and 2017. So it's 88,500. Yeah, it's easier to put them in the calculator without, you can't put million in the calculator, so you need to add 3 zeros. So it's really 1809000, and we want to do 1897500. Although I need to put that the other way around. Ugh, never mind. Anyway, you get the idea. Take the big number and take the small number away from the big number, and you'll get 88,500. And in these maths questions, you're going to get 1 mark for writing 1.809 million, or for writing the number of dairy cows in 2017. So before you do the subtraction. All right, let's move on. 2.4. Farming cattle for humans to eat is less efficient than farming crops because of energy transfer. So this kind of goes back to GCSE ecology, and you remember your pyramids of biomass. What happens at each energy level? What are the energy levels called in a pyramid or a food chain at A level? They're called trophic levels. So what happens between each trophic level? Why does the biomass decrease or the energy decrease between each trophic level? Of course, we get energy losses between each trophic level. So when an animal eats another animal, there may be undigested bits, bones, cartilage, hair they can't digest, so that energy is not passed on. The animal that's eating does excretion, so it loses energy like that. Respiratory losses, obviously breathing out carbon dioxide is a loss of mass and energy. Heat for warming your body, respiration produces heat. That is lost as well to the environment, or lots of it is lost to the environment. Movement, muscle contraction, all of these are plausible ways that energy is lost between different trophic levels. And this shows it would be more efficient in food chains if you cut down the number of trophic levels in your food chain. It is far more energy efficient. Anyway, 2 marks for that. Let's zing. Question 3. Oh my good gosh, what the arse is this? Look at the size of the stem. All right, nitrogen fixing bacteria such as Azobacteria corocum, whatever the hell that is, that binomial name, use the enzyme nitrogenase to produce ammonia from nitrogen gas in the air. Yeah, I'm struggling with the name of that, guys. Corococum can use ammonium, oh, if you must, can use ammonium chloride as a direct source of ammonia. When a source of ammonia is not available, the bacteria can use nitrogen fixation. A scientist investigated the effect of an increase in the concentration of ammonium chloride on the activity of nitrogenase in the bacteria. He prepared several liquid medium cultures of the bacterium. Each liquid culture had the same volume. He grew each culture in a different concentration of ammonium chloride. He recorded the nitrogenase activity in arbitrary units. He removed the bacteria and then recorded the concentration of ammonium chloride remaining in each liquid medium. We're going to be asked, apart from temperature and pH, give two variables the scientists should have controlled. So they already said they're using the same. I believe they said they're using the same. Well, no, we're changing the concentration of ammonium chloride, so we can't say that. But we should keep the volume of ammonium chloride solution the same. Also, what about the volume and concentration of the bacteria? You think about a culture of bacteria, it's basically a solution with a certain amount of bacteria um, per unit volume. So per centimeter cubed or per millimeter cubed or whatever. But you need the same rough amount of bacteria in each area. So the volume of bacteria, concentration of bacteria, that should be controlled. I can't remember, I'd have to flick up again on the question whether they said the volume of ammonium chloride, which does need to be controlled. They have changed the concentration, but if they mess around with the volume, that's going to affect the concentration. So we need to keep that the same as well. So there's all sorts of different things. Any two of these is going to grab you 2 marks. And there'll be plenty more on the mark scheme. Check the link in the description if you want to check out other alternative answers. 3.2, a student concluded that this investigation showed the ammonia inhibits nitrogenase activity in nitrogen-fixing bacteria. Use all the information to evaluate the student's conclusion. Remember, with evaluations, you're looking at for and against. So we're looking at what evidence is there in the data to say, the ammonia did inhibit nitrogenase. Basically the activity of that enzyme in nitrogen fixing bacteria. And then is there anything against that? So we can see here as the ammonium concentration increased, what happened to the enzyme nitrogenase? Well, the activity decreased. So straight away, that is a 0.4. And also as the concentration of the ammonium chloride remaining in the liquid increased, which means the nitrogenase is not breaking it down. So what we can say is 2 evidence points for there, which will score us 2 marks, and then we can just come up with 1 against. So as nitrogenase activity decreased, well, we saw nitrogenase activity decreased as the ammonium chloride concentration increased. That does suggest that the ammonium chloride is inhibiting the nitrogenase. Also, when the nitrogenase activity had decreased, at high concentrations of ammonium chloride, we got low activity of nitrogenase, but we also found that there were high concentrations of ammonium chloride remaining in the solution when the nitrogenase activity is 0. So those are points that are 4. Now, what we can also come up with, let's think of some against points. Normally it's things that they haven't done that's valid in the experiment. Well, the student concluded that the investigation that ammonia inhibits. Now, notice the student has said the word ammonia inhibits nitrogenase. What did they use in the experiment? They used ammonium chloride. Ammonium chloride and ammonia are not the same thing. So that's not true. Another statement is that, remember that really hard name that I couldn't pronounce, the cocochum, whatever the hell name it was? They only used one type of bacteria in this experiment. They used that Azobacter corococcum, when actually in the statement the student has made, the conclusion the student made, did not specify that species of bacteria. So they only did the experiment on one species. You can't label all nitrogen fixing bacteria. So that's going against this. And that would skew you the 3 marks. I'd also say the extra point is the experiment involved ammonium chloride, and yet the statement the student made referred purely to ammonia. And ammonia and ammonium chloride are 2 different things. Okay, 3.3, nitrogenase catalyzes the reduction of nitrogen during nitrogen fixation. The reaction requires 16 molecules of ATP for each molecule of nitrogen that is reduced. When ammonia inhibits nitrogenase activity, nitrogen fixing bacteria may benefit. Explain how. So even if you don't understand what's going on in this question, the key fact to this question is they're telling you that the reaction requires 16 molecules of ATP. Now, this reaction is no longer happening. How does that benefit the bacteria? Well, if the nitrogenase activity is inhibited, the bacteria benefit because instead of having to use those 16 molecules of ATP, um, fixing the nitrogen that's getting reduced, they have more ATP and energy available for other stuff. And you just need to give two examples of other things they can use ATP for and how they will increase the survival rates of the bacteria. So if they have more ATP, it means there's more energy available. That'll get us a mark. And explain what the energy could be used for. So it could be for growth, reproduction, for taking monomers and converting them into polymers, so constructing proteins from amino acids, all sorts of things the ATP can be used for. Also, they won't need to respire as much. A lower respiration rate will be needed. So, and there'll be more ATP available for phosphorylation. Phosphorylating is when you add a phosphate group to another chemical, which is what happens a lot in the organic chemistry within organisms. Do not say this means they can do more respiration. That's something that students wrote on this answer. Of course, ATP is made during respiration, so having more ATP doesn't mean you can do more respiration. Okay, the ATP is there's a net production of ATP in respiration. So yeah, and don't say mitosis, just say growth or binary fission. Mitosis is a form of asexual reproduction, but not for bacteria. Rolling, 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 rolling. Yeah, we're moving on now. 4.1, put a tick in the box next to the process that occurs in anaerobic respiration but doesn't occur in aerobic respiration. So basically, in anaerobic and aerobic respiration, does phosphorylation of glucose happen? Well, anaerobic and aerobic respiration both start with glycolysis, and you both kind of have the phosphorylation of glucose occurring in both of them. This question is actually asking us, where do you get a process that only happens in anaerobic but doesn't happen in aerobic? So, well, with the first one, glycolysis is in both, and glucose gets phosphorylated in glycolysis. And so I know that glycolysis happens before anaerobic and before aerobic. So you could either say phosphorylation of glucose doesn't happen in either, or it certainly doesn't happen in anaerobic respiration, but it does happen in glycolysis for both. So that's definitely not the answer. And then we've got the reduction of NAD. Does NAD get reduced in anaerobic but not in aerobic? Well, NAD definitely gets reduced in aerobic respiration, so that's not going to be an answer. The reduction of pyruvate, well, pyruvate enters the link reaction and actually gets carboxylated, I believe, in the link reaction. And so the pyruvate is going to end up forming like that acetyl CoA and that kind of business. So it's not getting reduced. However, in anaerobic respiration, the pyruvate molecule does get reduced in order to recycle the NAD reduced so that glycolysis can continue. The whole function of anaerobic respiration is to reduce the um, involves the reduction of pyruvate. Yes. Ta-da! Pyruvate does get reduced. And that is in anaerobic, but it doesn't get reduced in respiration. It's carboxylated in respiration. Okay, moving on. A student used the apparatus shown in figure 1 to measure the rate of anaerobic respiration in seeds for 48 hours. We've got a colored red liquid, I'm presuming it's red, normally is, in a graduated scale. And we've got a flask with some seeds in there and some potassium hydroxide solution, which absorbs the carbon dioxide, as we can see here. Now, the question says that this apparatus was left for 48 hours, and it says the red liquid moves to the left. Explain. So why does the red liquid move to the left? What we have to look at here is we've got some respiring seeds. So what are they doing when you do respiration? Well, they're taking in carbon dioxide. Taking in oxygen gas from the air within the tube. So they're absorbing oxygen. In fact, if we wanted to be fancy, we could say, ooh, they absorb 6 molecules of oxygen for every carbon dioxide, 6 molecules of carbon dioxide they release. So there wouldn't really be any change in the pressure in that tube if the carbon dioxide was swapped with the oxygen. However, the carbon dioxide is being absorbed by the potassium hydroxide. So we actually get a net decrease in the amount of molecules in the air in the tube. What does this do? This decreases either the volume, or you can say in the Mark scheme it decreases the pressure because there's less particles in the air. Because the carbon dioxide that's being released, which should be swapping for the oxygen in a 1-to-1, is being absorbed in the potassium hydroxide, and the oxygen is being taken in by the seeds. So we've got a net loss of particles in the air. That reduces the pressure in the tube, and the red liquid is sucked along that capillary tube there with the graduated scale. It's sucked along to the left down a pressure gradient from a higher air or atmospheric pressure in the air. You can see the end of the tube's open. So we've got air pressure there is higher than the pressure in the tube. So the red liquid is sucked along to the left. And that will get you your 3 marks. So let's smash that down into this answer here. Don't forget to hit the like, subscribe, share with other people, and check out my walkthroughs. I am aiming to get through every single walkthrough for all the exams for you. You definitely need to learn the content all off by heart if you want to get an A star, but you need to do all the walkthroughs or practice all the past papers as well. You need to be familiar with all the styles of question if you want to give yourself the best chance of getting that A star. So, yeah. This is a great way for measuring the rate of respiration. We can also measure oxygen consumption or a volume of carbon dioxide or rate of carbon dioxide produced. You can do all of that with this kind of apparatus and setup. Right, let's move on to 4.3, folks. Right, here we go. Zoom in a little. Apart from time, give 2 measurements the student would have to make to determine the rate of aerobic respiration of these seeds. So we know that you need time when you're calculating rate, but what other measurements should we do? Well, they want it, check out the units they've left us. Aerobic respiration, if you use your highlighter, this will trigger your memory. So always use a highlighter in the exam. Aerobic respiration of these seeds in centimeter cubed per hour. So if you need to work out the centimeter cubed, what do you need? To work out volume, what do you need? You need the length. Remember, this is in a tiny tube, so it's in a cylinder. How do you work out the volume of a cylinder? We're going to need the length of the tube, and we're going to need the diameter or the radius of the tube. So if you want to work out the volume of that cylinder, that's what we're going to need. And then we can do the rate or the volume of gas per hour. So always look at the units, always use your highlighter because that can have the answers in it right there. So we need the diameter or radius of the tube, the lumen of the tube, and the distance that the liquid has moved. Then we can work out the volume of gas produced or absorbed per hour. Awesome. Let's move on. Okay, so 4.4. The student used the same apparatus to determine the volume of carbon dioxide the seeds produce during 48 hours. So this time they want to work out how much CO2 is released during 48 hours. Give the change the student would need to make to the contents of the apparatus and describe how he could calculate the volume of carbon dioxide produced. So if you remember, we had that potassium hydroxide solution at the bottom that was absorbing the carbon dioxide. We need to remove that solution because we want the carbon dioxide to be released. And now what we could do is we could take the original set of results, so how far the red liquid moved with the potassium hydroxide solution, which removed the carbon dioxide, then repeat the exact same experiment again, but this time remove the potassium hydroxide solution and see how far the red liquid moves. The difference in the volume or the difference in the rate between those two will give us either the rate or the volume of carbon dioxide produced. So the difference in the volume produced in the first experiment when the CO2 was being absorbed to when the CO2 isn't being absorbed is the volume of carbon dioxide that is produced. In order to keep the conditions the same as a control, you would need to replace the potassium hydroxide solution with water. That way we have the same volume inside the tube and we don't affect the pressure, because remember what happens if you affect the pressure? The red liquid will move more. So we don't want that to be a factor. We want only the fact that carbon dioxide is being released. We only want that to affect the pressure. Right, 4.5. The student calculated that during the 48 hours, 6.2 times 10 to the minus 4 centimeter cubed of oxygen was absorbed by 40 grams of seeds. Calculate the oxygen uptake in centimeter cubed, g to the minus 1, hour to the minus 1. Now, you might not be studying maths and you might be like, what the hell is g to the minus 1, hour to the minus 1? It just means per. So centimeter cubed per gram per hour. Now, at GCSE, instead of writing to the minus 1, we just put a line, a diagonal line. So this is just saying centimeter cube divided by grams divided by hours. So calculate the oxygen uptake in centimeter cube per gram per hour. So this actually gives you, the unit gives you the answer. They've given us the volume of oxygen, which is 6.2 times 10 to the minus 4. We just need to divide that by the grams and then divide it by the hours. So we have 6.2 to the minus 4 centimeter cubed. You could either divide it by the hours first if you want, which is 48, or I think it's probably better to stick to the units. It won't make any difference to the answer, but just as an explanation to you guys, it's probably better that I don't divide it by 48 first. It won't make a difference whether I do 48 first or 40 first. But because grams come first, I'm going to go 6.2 times 10 to the minus 4 there is in 40 grams. So I'm going to divide it by 40. So 6.2 times 10 to the minus 4 centimeter cubed per gram I've now worked out. And that is 1.55 times 10 to the minus 5. So that's how much oxygen per gram, but that's over 48 hours. I now want oxygen per gram over 1 hour. So I'm going to divide by 48. So the units gave us the answer here. Take the volume, the centimeter cube, divide it by the grams, divide it by the hours. It was given in the answer. So divide by 40, divide by 48. And you get your final answer, which we can round to a suitable figure, which is 3.229. So we can round that. And it's 3.229 times 10 to the minus 7. So we can round that to 3.3 times 10 to the minus 7, or 3.33 recurring times 10 to the minus 7. Beautiful. And all that for 1 mark. Yeah. Don't you love A-level biology? Question 5. Yeah, whatever that was. Question 5. Lemurs are small mammals. I bet they are. Scientists use a computer program to predict the expected distribution of 2 species of lemur. I'm not even going to say them. Basically, let's call them E. rufus and E. rufrions on the island of Madagascar. These predictions were based on the environmental needs of each species. Then the scientists determined the actual distribution of these species of lemur on the island of Madagascar. So they used some sort of program to use the to predict based on the environmental needs of what these lemur species would need. They made a prediction about the distribution. And the actual distribution is showed with the little circles and the little black triangles. The actual distribution of E. rufus with the circles and the actual distribution of E. rufrions with the black triangles. And then you've got a river separating them. And then we have the expected distribution, what they expected in the shaded areas for each. Use figure 2 to give 3 conclusions you can make about the distribution of the lemur species. Now, when you see the word conclusion, this is something you can definitely say is true. So just stick to facts. Don't make judgments or estimations or predictions. Just stick to facts. And you can see, look at the expected distribution and look at the actual distribution. What can we say about where the actual distribution, where the little black triangles and white circles are, or grey circles are, compared to the shaded areas? Well, I can say that the actual distribution falls within the expected distribution in all points for both species. I can also say that the expected distribution is larger than the actual distribution for both species. These are 200% facts that we can say. So I could say E. rufus and E. rufrions, can't spell that, um, both their actual distributions fall within the predicted or expected distributions, okay, within the shaded areas. I can also make a second point and say that the expected distributions were bigger than the actual distributions for both. I can also say that E. rufus was found north of the river or to the northwest above the river, and I can say that E. rufrions was only found south of the river, or if I want to be funky, southwest of the river. Okay, so it was found in the southwest and southeast, wasn't it? The little black triangles were on both sides of the map. So I can 100% make a statement that E. rufrions were all below the river because none of them were found above the river. So there's loads of definite facts you can make about that map, and that's what a conclusion is, a pure fact that you can say, no judgment or, oh, I think this is because, no explanation, just what you say, what you see. Cool. And that was 5.1. So 5.2 says, using all the information, suggest how speciation happened to produce 2 species of lemur. So obviously at one point in time, there was just 1 species of lemur on Madagascar, and now we have 2 species. So, as with all types of speciation, we need to have a barrier that isolates the 2 populations, whether that's a physical barrier or a behavioral barrier or something else. Here on the map, we can clearly see there's a river. So, maybe the river provided the barrier, but it's definitely some sort of geographical isolation. And this eventually led to reproductive isolation. What does that mean? That means members of the 2 species cannot reproduce, okay, which means there's no gene flow between the 2 species. Now, over time, without that gene flow between the 2 species, the environmental conditions of the 2 geographical locations might be slightly different. So we have different selection pressures in each environment. Maybe one side is slightly warmer, the other colder, one side's damper, the other drier, different rocky terrain or trees in the other side, which means there'll be different selection pressures. And therefore, we get natural selection occurring due to random mutations causing variation and some alleles being selected for. So we now go into our natural selection bit. Due to the fact that some alleles will be beneficial and make those individuals with the beneficial allele more likely to survive, i.e., they can get more food, they can avoid predation, those kind of things. This will make them more likely to survive and pass that allele off to their offspring. And here's the A level bit of natural selection. We have to say increasing the frequency of the allele within the population. It's going to change the allele frequency in the population, normally by increasing that allele, that beneficial allele. Eventually, the 2 populations will have changed enough in their genetics that they will no longer be able to breed and produce fertile offspring. Even if you took 2 members from either side of the river, got them together, stuck them in a cage, and tried to get them to get it on, they would not produce a fertile offspring. They might at best be able to produce a hybrid. So now they are 2 species. Awesome. Okay, that's 5.2. Let's move on. Right. 5.3. The scientists used the mark release recapture method to determine the number of lemurs in one area of forest. They captured, marked, released a first sample of 30 lemurs. A week later, they recaptured a second sample of 25 lemurs from the same area of forest. The scientists calculated there were 250 lemurs in that area of the forest, so just one precaution needed when marking lemurs to make sure you estimate the number of lemurs is valid. Well, just out of a thing, the marking should be non-toxic. If you start killing the lemurs, then it's going to affect your population estimate. Also, mustn't be a fluorescent sign stuck on the back of their head, which lets all of the predators know where they are. So it mustn't affect their survival. It mustn't affect their chances of reproduction because that would affect the population or their chances of getting recaptured. I.e., if it makes it easier for you to recapture them, then it's not going to be a very accurate population estimate. Okay, 5.4. Use the information provided here in the stem to calculate how many lemurs were in the second sample that were actually marked. So we know what the total estimation of the lemurs was. It was 250. We know the first sample had 30, the second had 25, but we don't know how many in the second sample were marked with whatever fluorescent marking or whatever tag they'd used. So let's set the equation out and then rearrange it to find the answer. So the total population in the mark release recapture can be calculated by the number caught in the first sample, thus the 30, times the 25, the number caught in the second sample, divided by the number, can't spell, number of marked in the second sample, the number of lemurs that have the marking that we tagged in the second sample. Okay. Now, we don't know that here, but we do know the other figures. So we know the total population was 250. We know the number of lemurs was 30 in the first and 25 in the second. So we just need to do 250 equals 30 times 25 over x, solve x, and we have the answer. I mean, the big question is, could I have chosen a worse color to write this in? For those of you that don't have supernatural eyesight, I've written 30 times 15 over 250 equals x. So 30 times 25. So 30 times 25, I can't even read my own writing. 30 times 25 is 750 divided by 250. X is 3. So the number of marked lemurs in the second sample was 3. Okay, 6. In humans, the ABO blood groups and rhesus blood groups are under the genetic control. The inheritance of the ABO blood groups is controlled by 3 alleles of a single gene, IA, IB, and IO. The alleles IA and IB are codominant, and the allele IO is recessive to IA and recessive to IB. There are 4 ABO phenotypes, A, B, AB, and O. The gene for the rhesus blood groups has 2 alleles. The allele for rhesus positive R is dominant to the allele for rhesus negative lowercase r. The genes for ABO and rhesus blood groups are not sex linked, they're not found on the X and Y, and they're not linked or on the same chromosome. So we can't include them in Punnett squares together, that means. So 6.1, give the genotypes of the ABO blood groups for the individuals 1 and 2. So here we have 1. We can see that they are a rhesus positive male with the blood group A. This means if you have the blood group A, you can be IA or IAIO. Okay. The person with blood group 2 can only be IAIB. Codominant, they have one of each allele. So we know 2 is IAIB. But what is the blood group of, or what is the genotype of the phenotype blood group A for person 1? Well, we know that IA can be either of these two, but why can't they be IA? If they were IA, then we know that they couldn't make a blood group B number 4, right? Because they would have to give them an IA blood group. So that number 4 would have to be either IA, BA, or IA, BAB. You couldn't make blood group B on its own if person 1 was IA. So they must be IAIO, because that will tell us that the person blood group B there must have got the O from the father and the B from the mother. So yeah, there we go. A long way round, but they're IA. IO and AIB. Explain one piece of evidence from figure 3 that the allele for rhesus positive is dominant. People get have a crack up when I say allele because I can't pronounce it properly, but there we go. Okay, so what is the evidence that rhesus positive must be the dominant allele? Well, let's have a little look. Person 3 and 4, right? Blood group A and B, forget the blood groups. They're both rhesus positive. How did they have person 7, who's rhesus negative? If rhesus positive was lowercase r, then person 3 and 4 would be little r, little r, little r, little r. And what does that mean? That this person circled here, this child, would also have to be little r, little r. So it would be impossible for them to have, if rhesus positive was recessive and not dominant, it'd be impossible for two parents with homozygous recessive alleles to have any children other than children who are also homozygous recessive. So that kind of proves the link that proves this is positive, that this is dominant, is that 3 and 4 have to be heterozygous because they have produced 7 who must be homozygous recessive. So we know 3 and 4 are carriers of a recessive allele, which tells us that rhesus positive must be the dominant allele. And I like to go down the argument that if rhesus positive was recessive, then 3 and 4 could only have rhesus positive children. They couldn't have a rhesus negative children if it's recessive, because both the parents would be little r, meaning that how do you produce child number 7 who would have to have a dominant allele? Where did it come from? So we actually know that 3 and 4 are big R, big R, little r, and they produce 7 because the two little r's have come together. So that's the evidence that rhesus positive is the dominant allele, and you can choose whichever bit of that information you want to answer that question. Okay, good, sort of write up the answer here. So what you need to do is hit the like, subscribe, make sure you hit notifications, because I do live streams for A-level biology on a regular basis. I also do live streams for A-level chemistry on a regular basis. And anyone you know that's taking GCSE, share the channel with them, because it just helps build the channel. The more people that get involved and the more I can make the channel successful, the more walkthroughs and the more time I can devote to the channel. At the moment, it's a bit of a hobby, but I don't really have enough time to get all the walkthroughs done and all the videos that I want to get done for the channel. All right, nice one. Thanks for your support. Moving on to 6.3. Now that you have picked up your marks there, 6.3, calculate the probability of individuals 1 and 2 producing a rhesus positive son with blood group A, example individual 3. You can assume that individual 1 is heterozygous for the rhesus blood group. Show you're working. So here we're calculating the probability that 1 and 2, the two main parents here, are going to have another child like number 3. So a male that's rhesus positive. So with blood group A. So what's the probability of having another male child with the rhesus positive blood group and the A blood group? Calculate the probability. So really we've got 3 bits here. First of all, I want to find out what's their probability of them having blood group A. So we know that we've got, I haven't put the I's here, but we know we've got AB times AO. We know what the parents were. So we can work out, first of all, the probability of this child having blood group A. And the probability is they could either be AA or AO with the cross from the AB parent and the AO parent, which means the probability is 50%. So there's a 50% chance, or 0.5 if we want to do it in probs, that they have blood group A. Now I want to find out what is the chance of them being a boy or girl. You don't really need to draw this out, but I'm going to draw it out for you. You all know that chance. It's based on the chromosomes and the chance of the X and Ys or the XX coming together. So it's another 50%. We got XX crossed with XY in our Punnett square, and we're going to have a 50% chance or zero point 0.5 probability of them having a boy or a girl. So that is going to be 50%. So I now have a zero point 0.5 probability of A, a zero point 0.5 probability of boy. Now I need to write out the rhesus groups. So mum was rhesus negative, dad was heterozygous rhesus positive. This means they had a 50% chance of having a rhesus positive boy, so rhesus positive child. So we do 0.5, 0.5, 0.5, times them all by each other, get 12.5% or 1 over 8. So 3 Punnett squares, 0.5 times 0.5 times 0.5. You probably wouldn't have had to do the Punnett square for the genders. You know that one off by heart. Okay, 6.4, the frequencies, the scientists determine the frequencies of the ABO alleles and the ABO phenotypes in a large population. They then use a statistical test to determine if the frequencies of the 4 phenotypes differed significantly from the frequencies expected according to the Hardy-Weinberg. The frequencies of the IA-IO alleles were 0.15 and 0.65. What must the frequency of the IB allele? Right, you know Hardy-Weinberg is P plus Q equals 1. You can do 2P squared plus 2PQ plus 2 plus Q squared equals 1. But essentially, the main single alleles on their own have to equal 1. So we know P plus Q equals 1. Essentially, IA plus IO plus IB must equal 1. Well, if IA and IO are 0.8, then IB must be 0.2. There we go. They all have to add up to 1, the frequencies of the individual alleles. Name the statistical test you should use to determine if the observed frequencies of the 4 phenotypes differed significantly from the expected frequencies. Well, when you're looking at observed and expected frequencies, we use the chi-squared. And the number of the degrees of freedom for the chi-squared, well, what would that be? Well, we had 4 phenotypes, and degrees of freedom is the number of classes you're studying. So there were 4 phenotypes minus 1, n minus 1. We call it n minus 1. So we're going to take the 4 phenotypes and we're going to minus 1. So that's ABO and AB. We're going to minus 1 and we're going to get 3 is the degrees of freedom. So you take the 4 phenotypes as n. Degrees of freedom is always n minus 1 in chi-squared. So in this situation, degrees of freedom is 3. Lovely, bubbly. Right, 6.6. The scientists concluded the observed frequencies of the 4 phenotypes differed significantly from the expected frequencies. Use your knowledge of the Hardy-Weinberg principle to suggest 2 reasons why. If the Hardy-Weinberg principle is followed accurately, which is almost impossible, then the frequency of the alleles will not change from what is expected from population to population. However, there are about 7 assumptions we make with the Hardy-Weinberg principle, and you just got to name 2 of these. So it's like there shouldn't be any immigration or emigration, so nobody moving in or out of the population. There shouldn't be a high rate of mutation. So, for example, a high rate of mutation would be a reason why it's not appearing as the expected frequency. Mating should be completely random. Now, when is mating random? Okay, birds like to mate with the male who has the best song, or peacock with the best feathers. Mating is very rarely random, but the Hardy-Weinberg principle starts off on the basis that mating is completely random and not selected for. And there shouldn't be any particular selection of a particular allele. Shouldn't be like selected for either. So any of those can get you the marks. There are about 7 different assumptions you can make with the Hardy-Weinberg principle. Give 2 reasons why transmission across all right, we're on to 7. It's about neurons, obviously. Give 2 reasons why transmission across a cholinergic synapse is unidirectional. So check out my awesome GCSE D grade art here. The protein receptors or neurotransmitter receptors are only, the receptor sites are only found on the postsynaptic membrane. So you can only get depolarization of the postsynaptic membrane when the neurotransmitters connect there. The neurotransmitters can't turn around and can't connect to the presynaptic knob and depolarize that membrane. So that is one of the reasons why we only get unidirectional impulses. Another reason is that the vesicles containing the neurotransmitter are only found in the presynaptic knob and can only migrate there and diffuse into the synapse via exocytosis. There are no vesicles containing neurotransmitter in the postsynaptic membrane. So we can't have the neurotransmitter going the other way anyway, and even if they could go the other way, there aren't any receptor sites. So that is why the synapse ensures the unidirectional function of the nervous system, that the impulses, action potentials can only go in one way. Figure 4 shows the change in the membrane potential in the postsynaptic neuron after repeated summation from a single presynaptic neuron. We have the membrane potential on the left in millivolts. You can see the threshold's about minus 50. Until the threshold at minus 50 or minus 55 is breached, we don't get the action potential. We can see there's an addition of several action potentials here. We've got 1, 2, 3 action potentials, or whatever. We've got 3 lots of neurotransmitter, joining together. So over time, this is temporal summation because you've got the time at the bottom, and basically we're getting a combination of lots of neurotransmitter being released into the synapse over a short period of time, which is coming together as a summing together or coming together to actually cross the threshold and make an action potential. So we've got repeated impulses in a short time, releasing multiple amounts of neurotransmitter into the synaptic cleft, which combine to depolarize the postsynaptic membrane and form an action potential by crossing the threshold. Lovely bubbly, and that's known as temporal summation. The other type of summation, of course, is spatial summation. Okay, and that's to do with various different presynaptic knobs all releasing neurotransmitter at the same time. It's not to do with the time frame of the release of neurotransmitter, so shortly arriving impulses. It's to do with multiple postsynaptic knobs releasing their neurotransmitter, which all adds together to cause an action potential, basically to depolarize the postsynaptic membrane. So that's slightly different spatial summation. Okay. Keep moving on, keep moving on, keep moving on, keep moving on. Okay, 7.3. What the hell is that? Myasthenia gravis, MG, let's just call it, hey, is an autoimmune disease caused when antibodies bind to the sarcolemma, can't even say that, postsynaptic membrane of the neuromuscular junction. This can weaken contraction of muscles. So this autoimmune disease weakens the contraction of muscles by its antibodies binding to the sarcolemma, which is the postsynaptic membrane at the neuromuscular junction. Mestatin, however we say that, is a drug that inhibits the enzyme acetylcholinesterase. So we've got a drug that inhibits an enzyme. It can help in the treatment of MG. Suggest and explain how MG weakens the contraction of muscles. So how does the antibodies binding to the postsynaptic membrane weaken the contraction? Well, if the antibodies bind to the receptors, they're going to prevent acetylcholine from binding. What does that mean? If acetylcholine can't bind to the postsynaptic membrane at the neuromuscular junction, we don't get depolarization at the neuromuscular junction, and we get less action potentials. We can say less sodium ions enter the neuromuscular junction, so we get less depolarization. Less sodium gates are opened. And that's because the antibodies have blocked the binding site for the neurotransmitter, the acetylcholine. We're now asked, why can this drug help in the treatment of MG? Explain how. Well, what did it tell us the drug does? Well, the drug seems to be an inhibitor for acetylcholinesterase. If you inhibit acetylcholinesterase, they're essentially asking you, what does acetylcholinesterase do? Well, acetylcholinesterase breaks down acetylcholine in the synaptic cleft, so we don't get continual depolarization of the postsynaptic membrane. However, if we inhibit the enzyme acetylcholinesterase, then we're going to have more neurotransmitter in the synaptic cleft. So less neurotransmitter is broken down in the synapse, and we're going to have more neurotransmitter binding to the receptor sites, okay, on the postsynaptic membrane at the neuromuscular junction. They're going to depolarize that membrane, because they're opening more sodium gates, letting more sodium ions in, greater depolarization, greater chance of action potentials, or more frequent action potentials. So, really, that's how it works. And it means you're going to end up with more muscle contraction. So, it's going to help people who have very weak muscle contraction. Right, let's move on to question 8. All right, here we go. Scientists investigated the production of laboratory rats with the characteristics of type 2 diabetes. The scientists used the following method. They divided the rats into two groups, A and B, and fed them different diets for 2 weeks. They fed the rats in group A the normal diet containing 12% fat. They fed the rats in group B a high fat diet containing 56% fat. After 2 weeks, they injected both groups of rats with 35 milligrams per kilogram of the drug STZ, let's call it, to induce diabetes. 1 week later, the scientists determined the mean body mass and mean blood glucose concentration for each group. A value of plus or minus 2 times the SD from the mean includes over 95% of the data. Okay, so we have the kind of error bars or the standard deviation here written here, and what else? Calculate how many grams of STC should be injected into a rat with a mass of 230.45 grams. Show your working. Well, let's look at those units. We need 35 milligrams per kilogram. So first of all, um, let's turn the milligrams into grams, because our answer, they want it in calculate how many grams. Notice I highlighted that of STZ. So we need to turn it into grams. So 35 divided by 1,000 is 0.035 grams per kilo. Now, how much rat did we have? Did we have a 1 kilogram rat? That would be a massive fat rat. Okay. No, we didn't. We had a 230.45 gram rat. We need to convert that into kilos. So let's do 230.45 divided by 1,000. And we now get how heavy the rat was. And that's going to be like 0.23045, right? That is the kilograms of rat. So that's how heavy our rat is. And we needed 0.035 grams per kilo. So how much did we need for the this rat? Guess what? Times this by 0.35. So we just want to go 0.035 times 0.2304 kilos. If I had a 1 kilo rat, I'd times it by 1, and I'd need 0.035 grams for a 1 kilo rat. So how much am I going to need for a rat that's a quarter? I'm going to need basically a quarter of that. And we can get that by going 0.2304 times 0.035. And then we get our answer. And the answer is 8.064 times 10 to the minus 3. And they wanted the answer in standard form, so they needed it written like this, essentially. You could write, my gosh, you could write 8 times 10 to the minus 3 if you like. Right, let's move on. Suggest and explain why STZ was injected per unit of body mass. Well, the rats will all have different body masses. You can try to keep their body mass the same at the start of the experiment, but that's pretty hard. So you're going to need to inject them with a dose of STZ that is fair, basically. It needs to make a valid comparison between the rats. If I have a rat that's twice as heavy or twice as much mass, I need to inject it with a twice as big dose of STZ. So to make sure that they have the exact amount per unit of body mass. And it's because the rats have different starting masses. Okay, 8.3. The scientists concluded that Group B rats would be used for studying type 2 diabetes in humans. Use all of the information and your knowledge of type 2 diabetes to evaluate this conclusion. Remember, we want for and against. So, what they're basically translating this question, they're asking you, do you think the Group B rats are suitable for studying type 2 diabetes in humans? Ooh. give for and against. Is that 5 marks? If it is, you can do 3 and 2. Basically, you just need some points for and some points against when you have an evaluation. So when we looked at the results, we can see that the group in body mass B, um, the body mass increased in that group after their high fat diet. And it was significant because the SDs, the standard deviations, did not overlap, which means we have a significant difference in, it means their body mass increase was significant. And we know that obesity is a risk factor in type 2 diabetes. So that means, hey, this is a good conclusion here. Also, what else do we know? We know the mean concentration of the blood glucose, the standard deviations again did not overlap. So they had a significant increase in blood glucose concentration. Hey, guess what? People with type 2 diabetes have a higher blood glucose concentration. So these are really good reasons to say that this conclusion is a good conclusion. The group B rats could be used for studying type 2 diabetes. What can we say against it? They're asking us, can we use it to study type 2 diabetes in humans? Well, rats are not humans. Their anatomy and physiology is different. They are mammals, so it's a pretty decent comparison, but it's not the same as doing as making the statement about humans. Rats are not humans, so that would be against. Another 4 point that I've included there. They gave the rats a really high fat diet, and we know that a high fat diet is a risk with type 2 diabetes. So that's another 4 point. Another against is that the results were done over a short period of time. They were only done over 2 weeks. So we don't know the long term effects of the diet or the high blood glucose concentration or any of those things. They were not investigated. So we've got a couple of against points there, but loads of 4 points. All right. You needed any 5 of those, and they need to be distributed between 4 and against. 8.4, the scientists repeated the investigation using much higher doses of STZ. This led to the destruction of pancreatic cells. The scientists concluded that these rats would not be suitable for studying type 2 diabetes. Give 2 reasons why. Okay, well, when you destroy pancreatic cells, you can no longer release insulin. Well, type 2 diabetics still produce insulin, so this doesn't make sense to study people with destroyed pancreatic cells. Because they still produce insulin. With type 2 diabetes, it's actually that those people, their receptor cells on their liver and muscle membranes have become resistant to insulin. Pancreatic cells are not destroyed in type 2 diabetes. Okay, damage to the pancreatic cells may also affect other jobs of the pancreas, like its role in digestion and stuff that could affect the investigation. So lots of reasons why that's not a good comparison with type 2 diabetes. Question 9. Name the part of the body which releases antidiuretic hormone, ADH, into the blood. That's the posterior pituitary gland. Pituitary gland is GCSE knowledge. You will not get marks unless you say posterior pituitary gland. Of course, there is an anterior pituitary gland as well. Alcohol decreases the release of ADH into the blood, suggests 2 signs or symptoms which may result from a decrease in ADH. Right. If you don't have ADH, you don't get the aquaporins in the membrane. Less water is reabsorbed by osmosis in the collecting duct. So you're going to wee out a much larger volume of urine. You're going to go to toilet a lot more frequently, and you're going to be dehydrated. These are all symptoms. And your wee is going to be a lot lighter and more dilute. Describe the effect of ADH on the collecting ducts. I just mentioned that. ADH increases the amount of protein channels, which we can call aquaporins. It increases the number in the membrane of the collecting duct and the distal convoluted tubule, but we're focusing on the collecting duct here. And this increases the permeability of the cell membrane of the collecting duct to water. And what does that mean? That means that more water is reabsorbed from the filtrate into the blood by osmosis. Awesome. And that is the end of 9.3. Right, we're going into the last question now, guys. What the arse is this? Question 10. Read the following passage. BRCA1 and BRCA2 are human genes that code for tumor suppressant proteins. Mutations in BRCA1 and BRCA2 can cause cancer. Specific inherited mutations in these genes increase the risk of female breast cancer and ovarian cancer and have been associated with increased risks of several other types of cancer. Genetic testing using DNA from saliva can screen for all known harmful mutations in both genes. ER-positive breast cancers have receptors for the hormone estrogen. I can't bother to read all that. You're just going to have to pause the video, rewind, and read all of that, guys. Let's just crack on with the question. 10.1. BRCA1, BRCA2 are human genes that code for tumor suppressor proteins. Mutations in BRCA1 and BRCA2 can cause cancer. Explain how. So, mutation changes the base sequence. Get a lovely mark. This is going to change the primary structure or the order of amino acids, which changes the interaction of the R groups and therefore the bonding. And so it will change the tertiary structure, the 3D shape of the protein. It will now be dysfunctional or non-conformational. It will not be able to do its job. And the result of this misshaped protein is that it must result in rapid uncontrollable growth, which is the main cause of cancer, the formation of a tumor through rapid uncontrollable growth. And all of that comes down to a change in the primary structure, tertiary structure, 3D shape, and therefore leads to uncontrollable growth. Genetic testing using DNA from saliva can screen for all known harmful mutations in both genes. Describe how this DNA could be screened for all known harmful mutations in both genes. So we need to extract the DNA from the saliva, amplify it using PCR, use restriction enzymes to cut some fragments there, cut it into fragments, and then put that into do some gel electrophoresis, which will separate the different fragments based on their size. We can label them, we can mark them. You can do that using radioactive isotopes, or you can do that using fluorescence. And those markers, like probes, they join by a process called DNA hybridization. Now, we can identify them, right? We can identify them using fluorescent light or using radioactivity. And once we've identified them, we could compare them to like known bands, basically, of the fragments that cause these harmful mutations. So we could actually have known bands of the harmful mutations with how far they've moved in the electrophoresis gel, and that could all be labeled up, and we could find that in a data book, and we could compare our results to that. Effective treatment of ER positive breast cancers often involves the use of drugs which have a similar structure to estrogen. Suggest and explain how these drugs are an effective treatment of ER positive breast cancers. So if this drug has a similar structure to estrogen, that means it's going to compete with the estrogen to bind to the receptors. So the drug is binding where the estrogen would normally and prevents the estrogen binding. This means we get fewer transcription factors, basically like estrogen, can bind to the promoter region. And that means we're either going to get less or more transcription. Okay, 10.4. Blood tests can be used to test for cancers. However, the results of blood tests may not be conclusive when testing for prostate cancer. Explain why. All of these tests, whether you're doing the COVID test, the HIV test, you can have false positives. It also tells us in the text, just on line 11, it tells us that urinary infections or enlarged prostate can also cause high PSA concentrations, which is linked to prostate cancer. So it might not be that the person is suffering from prostate cancer. We can see here in the text, it might just be that they have urinary infections or an enlarged prostate, which also increases the concentration of PSA and will give them a positive result on the prostate cancer test. So, yeah. Right, let's look at the final part of the test paper. 10.5. Treatment with drugs might be able to reverse the epigenetic changes that cause cancers. Suggest and explain how. Epigenetic changes are generally caused by acetylation or methylation. I've got two little videos on that. You can see them in my description. So if you want to check those two tiny little videos out with little animations, you can find them in the description. So the drugs could affect methylation or acetylation. How? The drugs could decrease the amount of methylation on tumor suppressor genes, and this is going to allow for an increased rate of transcription. Okay, and that means we might get lots and lots of tumor suppressors coming out that stop us having tumors. Okay, so we want those tumor suppressor proteins to be made, and that can happen if we decrease the amount of methylation. Okay, that would get you all 3 marks. And there are some alternative answers as well. So if we increased, right, if we increased methylation of oncogenes, okay, then opposite to this is going to inhibit transcription. So increasing the methylation will inhibit the transcription or expression of these genes that can lead to a tumor. So you've got both answers here. You can either decrease methylation, and there's one mechanism where that can stop tumors, or you could increase methylation, and there's one mechanism where that could stop tumors. And you've got the same answer, you could say, with acetylation of the histones, where it stimulates the transcription of the tumor suppressor genes. The acetylation of the histones opens the DNA out, allowing transcription machinery, such as RNA polymerase, to get in and transcribe those genes. If you decrease the acetylation, the DNA becomes more tightly wound around the histones, and you're going to decrease the amount of transcription of certain genes. Now, you don't want to decrease the tumor suppressor genes. They're the things that are stopping you developing a cancer, but you might want to decrease the transcription of oncogenes. And there you go. That is the end.
You're still here? It's over.